Hello, and welcome to what I'm calling a super recap, which is trying to pick out some of the most important things that you need to think about regarding the seaweed physiology lectures. In particular, looking at things that I think you really need to know. And I'm going to be very quick here. You can always go back and look at the earlier videos illustrating some of these more important concepts. The first one is the complexity of life histories of different species of seaweed. So you have single morphological phase where everything is the same, haploid or diploid approaches, or you can have a haploid-diplontic approach that contains two morphological stages. Remember that there are isomorphic species, ones that the two stages are physically identical, same environmental requirements, but will have different reproductive structures and may have different cytology. They may be haploid, they might be diploid. You also have the heteromorphic species, those which have very different sporophytes versus gametophytes, such as laminaria or porphyra. The red algae, due to the lack of swimming sperm, have evolved a more complex life history that includes three morphological stages, including a tetrasporophyte that allows them to maximize the chances of establishment, settlement, and so on, following a single fertilization event. Recall the few called life history, complex, containing a main structure circling around an adult alga, producing eggs and sperm, forms a propagule, zygote, propagule germling, turning into an adult. Okay? Macroscopic versus microscopic levels. The more complicated kelp life history example indicates that two morphological phases exist. The microscopic gametophyte that you won't be able to see against the macroscopic sporophyte that is the large brown algae that are found around the shorelines of the UK. Chondrus crispus represents a really neat example of the complex red algal life history that incorporates the three morphological stages, including carposporophyte, tetrasporophytes, and the tetrasporophyte producing 4 spores for every single fertilization event. Recall how seaweeds is established, the varying ways at which they settle, attach, leading to establishment and growth of the adult alga. The stress concept. Not only are we talking about stress, we're talking about severity, duration, the frequency of exposures, and the additive effect of combinations of stresses. Think about how they can resist or just become susceptible to the change. Do they adapt? Do they conform? Do they regulate? Think about how light affects the growth of species, the effect of canopies driving the ecology of environment. Remember the PE or PI curve. Think about the light environment, what adaptation the species might have that allows it to maximize its growth rate, photosynthetic rate, oxygen production, carbon dioxide consumption. Think about the specific examples of shade adapted or light adapted species, the size of the photosynthetic array that allows them to capture more light but may lead to photochemical quenching that reduces the energetic requirement by dissipating energy. Think about how different stressors such as salinity and salinity, light, and temperature interact. Think about how algae grow faster at different temperatures that give them quite clearly defined thermal ranges. Think about the stochastic events that lead to loss and mortality of algae. Think about how algae cope with salinity that's variable in the local environment, the rates of change that it can undergo, the transportation of ions, the synthesis of organic solutes that allows them to adapt to these conditions. Think about DMSP, vitally important chemical that can be utilized for a variety of roles in many algal species around the world. Think about resource allocation, the packets of growth, reproduction and defense that can be used to model how organisms respond to change as well as to differential pressure. Think about optimal defense theory. The allocation of energy into different parts of the body or frond of an alga. Think about the theoretical context of grazing pressure in algae and invasion success, how this whole interplay of this arms race between grazers and the algae come together to lead to these models of invasion dynamics. In the past, I have asked exam questions which were about how seaweeds respond to light, temperature, salinity and so on. This year I want people to think more about the ecological context, how it all fits together to drive how seaweeds establish and colonize the shores. The lessons learned here are important not only for macroalgal ecology and physiology, but also for wider ecology and wider understandings of how organisms respond and adapt to a variety of different stressors and different environments.